right, so now we're going down into the rabbit hole. Don't be afraid, Alice. We're talking about going down into the muscle cell and talking about how those muscle cells do their contraction. So where we left off? Probably weren't laughing. You may be at the structural complexity of all this, but when we last left off, we were talking about how the neuron comes down and stimulates the muscle cell at that motor end plate, and how it basically transfers a signal, right? We saw an action potential coming down the axon, then we saw that electrochemical signal change into a chemical signal as the acetylcholine, the neurotransmitter, was released across the synaptic cleft and was binding with receptors that were found on the motor end plate of the muscle cell. Okay? So now what we're going to be looking at is, okay, now that the signal has gotten to the muscle cell, how does that spread? And how does it get down into where all of those myofibrils are and where those actin and myosin filaments are? All right, so when we're starting out then, we're starting out with the sarcolemma that's been stimulated, right? And it's sitting at what's called a resting potential. And we mentioned a little bit about this when we were talking about the plasma membrane or the cell membranes, and how there's, in most cells, an uneven distribution of charged particles on either side. All right, so in most cases with these cells, the inside of the cell is relatively negative compared to the outside of the cell. So what we're going to find is we are going to go from a resting potential to an action potential, which is the signal that's then going to spread along and into the muscle cell itself. So we start out with what's called an end plate potential, which is, like we said, that area of the muscle cell that's right underneath where that synaptic end bulb is sitting. So along here we had chemically gated channels, right? Chemically gated or ligand channels are going to be binding that acetylcholine. So these channels bind the acetylcholine. Oh, that was a bad line. These channels are going to be binding the acetylcholine. They open up like you saw in the animation, and the sodium goes in. And so what happens there is that motor end plate and that area of the sarcolemma is going to become less and less negative, because we have positively charged sodium ions, cations, that are going into the cell, right? So if in your life you start getting all these positive things, right? You get a raise at work, and all of a sudden you meet somebody new or something like that, right? Whereas you were negative, now you become more and more positive. So this is a change in what we call the end plate potential. So that's what we see here, is how, again, those neurotransmitters are coming across. They're binding to little channels across here, opening up, allowing sodium to diffuse in, and we're going to get a end plate potential right there. So what's going to happen then is we're going to depolarize the membrane, right? So voltage gated sodium channels are going to be involved. And so what's going to happen here is, right, channels need a stimulus as to when to open and when to close, just like a muscle cell needs a stimulus when to contract and when to relax. So we're looking at chemically gated channels. They needed to bind a chemical that tells them when to time it's open. These are called voltage gated channels. And if there's enough sodium that's flowing into the cell and the cell becomes depolarized enough, it will reach threshold that will cause these voltage gated channels to open up. It's a bit like if you have a like a gate at your community or something like that, and somebody has to call, and they call you up, and then you send a signal, right? You send them a bzzz, you send a signal to the gate, and that opens up the gate, right? An electrical signal. So that's what we're going to find is becoming involved here. So we have voltage gated channels that are in the adjacent area, and this one right here is a sodium channel, a voltage gated sodium channel. And so once that charge that we see in the membrane right here reaches threshold, a particular, like in, we talk about like cardiac muscle and neurons, right? Those are excitable cells, skeletal muscles, etc. So like in neurons, it's about negative 55 millivolts, but for different cells, it's different. But when it gets to that level, then that's the stimulus for these channels to open up, leading to more sodium coming in, more depolarization. So we're going to generate this depolarization where the inside of the cell gets less and less polarized. It goes from, being down at like negative 70 millivolts, up. It's getting less and less negative. And then, just like things that go up, they have to come down. So essentially, when this action potential is generated, we're going to depolarize, and the curve's going to go up, and it's going to repolarize, and it's going to come down again. And the repolarization comes from potassium channels instead. So potassium channels, what happens is, after the sodium channels open and let sodium in, they close, and then it's the potassium's turn. And potassium channels open up, and we have a high concentration of potassium inside the cell relative to outside the cell. So the potassium is going to diffuse out of the cell, right? And so now we have a cell where we're losing positively charged ions out of the cell, making it more and more and more negative again. So what we find is that an action potential tends to look like this. So here's our resting potential right here, right? So our resting potential is about negative 90 millivolts. Again, it's different for different cells, but this is when the muscle cell is not getting stimulated. It's at rest. Now, neurotransmitter voltage-gated channels to chemically gated channels, spreading to voltage-gated channels, and now more and more sodium channels are opening, and that's going to cause depolarization of the cell, right? So it started at negative 90 millivolts, and it's going up and up and up towards zero. And when we get towards zero, right, that's when the cell wouldn't be polarized anymore. So when we're going from negative up towards zero, we call that depolarization, even as it goes up to like plus 30 millivolts. Then repolarization, like we said, sodium channels close, potassium channels open. Now the positively charged potassium is leaking out of the cell, so that brings it down again in what's called repolarization. So that's, to a certain extent, what an action potential, at least graphically, looks like in terms of the change in membrane potential. We'll see that again when we get to neurons. We'll see that again when we get to the cardiovascular system and how it may be similar or different to this. Okay, so what we're doing is we're getting this cell excited, right? We're getting this muscle cell, it's receiving the signal is spreading. And so it's spreading along the surface, but it has to get deep inside of the cell as well, where the myofibrils exist, where the actin and myosin are as well. So what we find is that the action potential is going to go along the sarcolemma, but remember we have these invaginations as well that dip into it, which are called T-tubules. And the T-tubules go down, and right associated with the T-tubules are little sections of sarcoplasmic reticulum. Remember, in the muscle cells, the sarcoplasmic reticulum holds calcium, so it's going to start releasing calcium. And just like we saw calcium before in the neuron, acts as a second messenger. This is an intracellular messenger now. Right? So this is where we were focused. This is where the signal was starting, and then it's going to spread, like we see it spreading along the surface here, and then we see the T-tubules going inside of the cell. The sarcoplasmic reticulum associated with it, and all the calcium flooding in amongst all these different myofibrils like we see here. Now inside the myofibrils, right, here's a T-tubule, it's coming in like this. Here's the sarcoplasmic reticulum releasing the calcium. Here's the calcium going down and interacting at this junction between the actin and the myosin. Now remember, we said actin and myosin are the contractile proteins that are doing the sliding, but we said troponin and tropomyosin are the management or the regulatory proteins. And in fact, that calcium goes down and it binds with the troponin, and it moves the troponin and tropomyosin out of the way so that the actin and the myosin can interact. So, again, lots of times, it's kind of hard to picture how that is occurring when we're just looking at something that's a static image. So let's see if it helps if we look at this animation.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
So it's kind of interesting to think about that, right? So when we were going through skeletal system, we were talking about homeostasis and what the skeleton does, things like that. We're talking about calcium reserves. And remember how we had osteoclasts that would chew up the bone and osteoblasts that would build the bone? Well, on the one hand, you're like, why would we ever want to chew up bone? That doesn't make sense, right? You're going to get osteoporosis or something. But here you're seeing two different critical systems of the body that use calcium all the time, right? Both the nervous system and the muscular system. And they need stores of calcium to be able to get excited, to send messages, to contract, etc. Okay. So, basically, calcium is what is telling the muscle to contract. And so, like we said, it's going to involve those regulatory proteins, and then it's going to allow our different contractile proteins to interact. So we're going to see that coming up in the whole event that we call sliding filament mechanism. Okay? Now these steps that you see here are something that we're going to go over one by one as we're understanding how all of these different proteins interact and calcium roles in it. But this is the text, and we're going to come up to the dynamic access. So the access of the calcium coming from the sarcoplasmic reticulum, getting access to the troponin, and then causing that troponin and tropomyosin complex to move out of the way. So remember, oftentimes the sliding filament mechanism is also referred to as this idea of the cross-bridge cycling, because that myosin head makes a bridge with the actin molecule. So first step, cross-bridge formation, then we have the power stroke, then we have the cross-bridge detachment, and then we have the cocking of the myosin head. So here's the deal, something to remember. I like to think always in terms of analogies, and so if we're talking about the actin molecule or the actin thin fiber with the actin molecules all along it, right, and the tropomyosin coiling around it and the troponin every once in a while, that's kind of like the rope in a tug of war, and the myosin molecules with their cross-bridge heads are grabbing onto it just like you would grab onto that rope in order to pull it past you in order to move the other team closer and closer together. So when you're thinking about that, if you're thinking about doing that type of an activity at the picnic or whatever, right, the tug of war, you can't just grab onto the rope and do nothing, right? That's only the first step. It's definitely a critical first step, grabbing onto the rope, but that doesn't win that contest, right? So you grab onto the rope, you pull, right? That's the power stroke. And then what you have to do is you have to release at least one hand in order to get some power back again, grab onto the rope in the next section, then release and grab and pull, release and grab and pull. Hand over hand action, right? And when we look at those thick filaments, we've got lots of these different myosin heads, and they're doing that type of thing. So they're not all contracted at the same time, just like your hands and the hands of the people on your team. Grabbing onto that rope at different times and pulling it along is going to get that thin fiber to slide. Okay? Okay. So this view that we see right here is showing the actual interaction of these different protein components. So here in the middle we can see a thin filament, right? Shown in blue right there. And we can't quite see the thick filament in this depiction. But the thick filament with its cross bridge would be like coming across like this, and across like this, right? On either side of the thin filament. So if we zoom up on that area and we're looking kind of down the barrel of it, We can see the actin molecules right here as part of the thin filament. We can see the troponin complex, this yellow one right here. And we can see the tropomyosin. Now here's the key thing. Here, right there in red, right? That's the myosin head. That's the cross bridge. And it wants to come forward and interact with and bind with, grab a hold of that rope, which we call actin. But the troponin and the tropomyosin are in the way. And so what we have to do is we have to move those out of the way. And remember we talked about that calcium coming in? So that calcium is going to come in. It binds with the troponin complex, which causes this whole complex to move out of the way, pulling the tropomyosin with it. And now, look, we're not blocked anymore. We're not physically blocked anymore. The myosin and the actin are going to be allowed to interact. Okay? So we'll see that in a view that's a little bit more similar to what we've been looking at before. So here we see, and again, this is taken from a different textbook, but I thought it did a good job of really zooming up on this interaction of the thin filaments in the center with the thick filaments on either side of it. Now again, you can see how the purple in this case, cross bridges or heads are all around. So they're going to be interacting with a bunch of different thin filaments. But here's our first step that we see right here, right? This is the resting sarcomere. So notice that the cross bridges are disengaged, right? Like when you start a tug of war, you're not going to be holding on to the rope, right? So they're disengaged and they are kind of recocked, right? They're all jazzed up. They have this potential energy inside and they're working to make it actual kinetic energy. So if you look in the head, you may recognize this, right? What that is, those are the broken components of an ATP molecule, right? Hydrolyzed ATP, we liberate that energy from the ATP molecule, and now we've got energy inside of these heads. So they're just waiting for their chance, right? And right here, at least between the thick and thin filaments, that signal from the calcium, from the surface, coming down inside and causing the calcium release has not occurred yet. All right, so let's go to our next step. So our next step here is that we get an action potential coming from the motor neuron. It goes along the T-tubules deep inside, and it causes the release of calcium from the sarcoplasmic reticulum. The calcium floods in here, okay, and they have it just as a box right here, and it binds to the troponin, right? That three-part molecule that we see right there in yellow represents the troponin. So it binds to the troponin, and because the troponin and the tropomyosin are connected, when the troponin changes shape, it pulls the tropomyosin with it. Now, one of the analogies that I like to think about in terms of this interaction is like how the thick filament and the thin filament almost are like lovelorn teenagers, right? And so they're in the house and they would like to get together physically, right? But the parents are thinking, no way. Not in my house, not under my roof. And so the parents, the troponin and the tropomyosin, are physically in the way. Imagine them on the couch, right? There's the actin, there's the myosin. They really want to get together. And then you have the troponin and the tropomyosin physically between them, keeping them apart, right? But then what happens, in the analogy anyway, is maybe you have a friend or a sibling or something like call up the parents and say, oh, can you come get me? I got stranded over here. My friends left me, whatever. The parents have to leave. And as the parents, the troponin and the tropomyosin complex move out of the way, oh, now the actin and the myosin are able to get together and get physical, right? So calcium comes in. That's kind of the phone call coming in, causing the parents to leave, causing the regulatory proteins to leave, and now our actin and myosin are going to interact. So you can see it right there, right? See how they're kissing. Ooh, right there, the active site on the actin. But you can see how the myosin heads come out like that, right? So they come out like that, and then the next step, you know, where we were before, we were just connecting, like we said, grabbing the rope of a tug of war. But the next most important step is this power stroke. See how we had a ratcheting? It connected kind of over here, and then it moved over that way. So it moved, and so that ratcheting, like this, grabs onto the grabs onto the thin filament, and then pushes it ever so slightly, just like you grabbing onto the rope and then pulling. And then, right, if you're going to continue, you're going to need some more energy in order to grab again. So we see the components of the broken ATP molecule disengaging. And what we're going to do is we're going to get some new energy. A new ATP molecule is going to bind with the head. That's going to cause it to release. So then now it has the energy to go back and grab it again. So, you see how it's gone back to its original cocked position right here? Okay? So if the calcium levels remain high because the cell is getting stimulated, then the parents are going to be out of the way, right? As long as that sibling, that younger sibling needs them, they're going to be out of the way. So if the calcium levels remain high, then the troponin and tropomyosin molecules are pulled out of the way, and the actin and the myosin keep interacting, right? But if the calcium levels drop and it gets taken back up into the sarcoplasmic reticulum, that emergency that called the parents away is all done. Now they come back into place, physically get there on the couch and get in between that actin and myosin. Okay? So it's the series of events that we just depicted that causes the muscular contraction to occur again and again and again, shortening the sarcomere, shortening that myofibril, shortening that actual muscle cell and muscle itself. So if you look at it, it tends to look like this, right? So that's one myosin head interacting with the thin filament. We can see the actin in orange there. You can see how, when you look at it, right, what's dropping in there? You see that yellow coming in from the upper right-hand corner? That's the calcium coming in. It binds with the troponin, shows in purple. That moves out of the way, pulling the tropomyosin with it, and that allows the myosin head to drop in and interact. That little cone that you see there as well is magnesium. Also important, but we're focusing on the other components instead. Right? So see how that rope is moving on through. It's just one myosin head, but many of them grabbing on at different times and resetting are going to slide that filament past the other. So it starts out something like this, right? All open like that, but then through time, it's going to do that, right? And you can see how those thin filaments and thick filaments the thin filaments are sliding past those thick filaments like that, right? They're not shortening, but they're sliding past one another. So that's important in terms of this sliding filament theory. It was developed by a guy named Huxley in 1954. Good, like 70, almost 5 years ago now. Pretty amazing, rather than just mice running underneath the skin, right? Okay. So let's take a look at a little video to try to cement it, because this is a critical, like we said, this is a critical part of knowing how the muscular system works. This is the crux of it. All of the different muscles that we see around the body that we're going to learn are doing this. So if you don't understand this, then you don't understand how the whole system itself works.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments.
See, it looks like a tug of war, doesn't it?
The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released. and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
And there you go. So, I know it's a lot to take in, a lot of steps, but you need to know those key players. Actin, myosin, troponin, tropomyosin, calcium, and ATP. You need to know all 6 of those players. You need to be able to describe what they do to allow these muscles to shorten, to allow these filaments to slide past one another. Okay? All right. So we're going to stop here. Let your mind kind of come back into place after it's been exploded. And then we'll touch base again and continue to talk about the intricacies of muscle contraction. Now that we know how it works on an individual cellular basis, we need to talk about how those cells perform, etc. We'll do that next.